Okay, friends, we have studied in sexual reproduction in flowering plants so far. The very first step. This was the first step in which we have studied how the gametes are produced, what are the structure of male reproductive system and female reproductive system, and today we will talk about the second step, which is fertilization event. Actually, this is called double fertilization event, and this is a very big question for your board examination. Why it is called double fertilization? So today's topic will be double fertilization. In first two videos, I had explained about male gamete and female gamete. Before that, I need to introduce some topic here to you people. Like if I draw a female reproductive system, this is a female reproductive system, and as I told you in the very beginning, this is called stigma, style, and this one is ovary. This is called pistil. This is also known as carpel. This is a structure of female reproductive structure, and this is the structure which we have to understand. This is the structure of a pollen. This pollen is made up of two types of cells. This is generative cell and this is vegetative cell. These are the two cells with a mature pollen. 60%, this is the structure of pollen. 60% of pollen grains, they are made up of two cells in angiosperm, and remaining 40% of pollen of angiospermic plants, this will be three-celled structure, which we don't have to study here. So this is to be transferred here to the stigma. It goes into stigma. There will be some ovules into it, maybe 1, 2, 3, and it strikes here with the help of air, pollination by air, with the help of an animal, pollination by animal, with the help of an insect, pollination by insect, with the help of water, pollination by the water. By several means it can enter, it can strike on the stigma, and it makes a pollen tube. Through the pollen tube it reaches into the female gamete present here. Female gamete present here inside the ovule. Right? So inside the ovule, female gamete is there, which has to be fertilized. So fertilization we have to study, but this, what I said, double fertilization, this means a lot for this topic today. So what is the gamete? The male gamete I have made here, the female gamete we have to make here. Let's draw the diagram of female gamete. This is a big cell. I hope you understand. The diagram I'm drawing is embryo sac. It has three cells in a row. One, two, three cells with their each nucleus. Two cells here. Two cells, and inside the cells finger-like projections are there. Finger-like projections are there, and just behind it there is another cell which is called egg cell. So it has its own nucleus, it has its own nucleus. So these all three have their own nucleus into it. The important thing is leveling and this name also. And in the middle of this, two nuclei will be there, two nucleus in the center. So this is what I have drawn, an embryo sac. This structure, remember it students, this structure is very important for your board examination or anywhere if you go, and if you are going and studying botany or the biology, here we have to discuss for sure the embryo sac if we are studying the sexual reproduction. So this is the female gamete present here, what we have to study today, and the cells are here. So I hope you understand how many cells are there. Let us level first. These are called antipodals. These cells are antipodal cells or antipodals. These two cells, similar in structure and having finger-like projection in it, these are called synergids. So these are synergids. Finger-like projections are there, which I have shown in green in color. These are called filiform apparatus. Filiform apparatus. And the cell which I have drawn black in color, it is called egg cell. And two nuclei in the central cell, in the middle of central cell, these are called polar nuclei. These are called polar nuclei. So again we have to count, we need to count the number. Antipodals are 3 in number, egg cell is 1 in number, synergids are 2 in number. So total number of cell, 2 plus 1 is 3 plus 3 is 6, and the bigger one 7 cell. So this embryo sac is also called 7 celled. And how many nuclei are there? 1, 2, 3, 4, 5, 6, 7, 8. 7 celled, 8 nucleate structure. So we have seen 7 celled 8 nucleate structure. It means that this embryo sac is also called 7 celled 8 nucleate structure. So this is the structure of an embryo sac, and this is male gamete. Now what happens if a pollen is striking over the stigma? It makes a pollen tube, and the pollen tube will goes down towards the embryo sac is present inside the ovule in the ovary. So ovules are there. The structure I have made. Let me draw once again the structure of ovule for your better understanding. These are outer epidermal layer, so many layers, nucellus into it, and inside it here, how safely it is present into an ovule. Ovules are there, 1, 2, 3, maybe one or more than one. So this embryo sac is present. The embryo sac is present inside the ovule, but I have drawn here. So once it enters, it is entered by micropyle. This end is called micropylar end, and this is called chalaza end. So it is entering, the pollen is entering. Remember, pollens have two cells, vegetative cells and generative cell. So this, now listen to me carefully, it's very damn important thing is this, what I am going to teach you. The pollen will start entering into it, and the entry will be facilitated by filiform apparatus, the finger-like green color structure present in the synergids. It facilitates the entry of pollen into it. So the entry will be done. Two cells are there. So generative cell, this generative cell, here the small one will fuse with egg cell. Egg cell. Fusion of generative cell with egg cell, and that form zygote. That form zygote. But one more cell is remaining left. Generative only fuse, na? This vegetative cell is remain left. So this vegetative cell will go and fuse to polar nuclei. So we can write another box. Vegetative cell fuse with polar nuclei, and that form primary endosperm nucleus, and it forms primary endosperm nucleus. I just want to tell you, fusion is happening with these two cells to the two cells of embryo sac. Generative cell fuses with the egg cell. Generative cell fuses with the egg cell to form zygote. Vegetative cell fuse with polar nuclei, these two nuclei, and form primary endosperm nucleus. Students, it's a very important topic. I'm telling you many times, I have said you it's very important topic. Literally, I'm telling you so many questions will be there. Many questions are waiting for you in the boards from same topic. So listen to me carefully again. Zygote and pen. Primary endosperm nucleus has been developed, is been formed. Now what will be happen? This zygote will developing, and this primary endosperm nucleus, let me write name. Primary endosperm nucleus that develops into primary endosperm cell. This develops into primary endosperm cell, and this primary endosperm cell will be consumed in the development of zygote produced by the fertilization between generative cell and egg cell. So the function of primary endosperm cell is to nourish the zygote. For the development of zygote, primary endosperm cell is required. Okay, so we can see in this complete process, fertilization is happening with egg cell. Egg cell is fertilizing with generative cell, and polar nuclei fertilizing with vegetative cell. That is why this process is called double fertilization, and fusion is happening between these two cells and one cell of this vegetative cell. So fusion is happening in between three nuclei. That is why this complete process is called double fertilization and triple fusion. And this is the complete process of fertilization. Now let me go. I'm going to explain it in Hindi as well for your convenience. As I told you, this is the female structure and this is the female male gamete. This male gamete strike, ये male gamete strike करता है स्टिग्मा पे, और ये pollen ट्यूब बनाते हुए वो इसके अंदर प्रेजेंट जो ओव्यूल है उसके अंदर गैमिट्स होते हैं, फीमेल गैमिट्स, उससे फ्यूज करता है. सो फ्यूजन कैसे होता है? इसका जो जेनरेटिव सेल होता है वो फ्यूज करता है एग सेल से, और इसका वेजिटेटिव सेल इन दो न्यूक्लियाई से फ्यूज करता है. इसीलिए फ्यूजन दो बार हो रहा है, फर्टिलाइजेशन दो बार हो रहा है, इसीलिए इसको डबल फर्टिलाइजेशन कहते हैं, जैसा कि मैंने यहाँ लिखा है. और फ्यूजन यहाँ पे दो न्यूक्लियस यहाँ ऑलरेडी थे और एक न्यूक्लियस वेजिटेटिव सेल का, तो तीन न्यूक्लियाई फ्यूज हो रहे हैं, इसलिए इसको कहते हैं ट्रिपल फ्यूजन. सो दिस विल बी कॉल्ड ट्रिपल फ्यूजन, और इसीलिए इस प्रोसेस को डबल फर्टिलाइजेशन ट्रिपल फ्यूजन कहते हैं. सो दिस इज ऑल अबाउट द डबल फर्टिलाइजेशन एंड ट्रिपल फ्यूजन. अनदर थिंग, प्राइमरी एंडोस्पर्म, ये जो पोलर न्यूक्लियाई है, इसका फ्यूजन जब वेजिटेटिव सेल से होता है तो ये वेजिटेटिव सेल फ्यूज करता है पोलर न्यूक्लियाई से, तो बनता है प्राइमरी एंडोस्पर्म न्यूक्लियस. यहाँ पे लिखा है मैंने प्राइमरी एंडोस्पर्म न्यूक्लियस. जो प्राइमरी एंडोस्पर्म न्यूक्लियस डेवलप होकर और प्राइमरी एंडोस्पर्म सेल में कन्वर्ट हो जाता है, जो यूटिलाइज होता है इस जाइगोट के फॉर्मेशन में. So I hope you all enjoyed my YouTube channel Ravi Sir Biology class, and I will always try to make the topics very easy for my dear students. Thank you.